Endonucleases cleave DNA internally and do not require free DNA ends for activity. This is in contrast to exonucleases, which require and act on free DNA enzymes that catalyze the removal of nucleotides in either the 5' to 3' or the 3' to 5' direction from the ends of single-stranded and/or double-stranded DNA. Removal of nucleotides is achieved by cleavage of phosphodiester bonds via hydrolysis. These important enzymes
Question might come in your mind while studying about restriction enzymes that if a bacterial cell produces these enzymes, then how can they protect their own DNA from being cut by the restriction enzymes? The fact is, restriction enzymes can't cut own DNA as there is a system called restriction modification system. Specific enzymes are present in that bacterium which modifies the restriction sites so that the restriction enzymes can't recognize it. Functional in nature and have two different subunits. It can cut the DNA at 24 to 26 base pair 3' to restriction site. Example, ECO, P of these recognition sites are with palindromic sequences within the DNA duplex. Well, palindromic sequences are those sequences which read the same from both the directions. That means the two strands of DNA run the same nucleotide sequence but just on the opposite direction, like this. By cleaving, the restriction endonucleases cut both the strands of DNA as the same recognition sequence present on both the strands. It produces staggered cut, generating DNA fragments, each with single-stranded complementary linker at either end. And as a result, a single-stranded DNA fragment is formed on each end, which is a few nucleotides long. They are called single-stranded DNA linker. The single-stranded DNA linkers are complementary to each other. These are called the sticky ends. So the sticky ends can pair to each other and can join together by the help of DNA ligase enzyme, which helps in reforming the phosphodiester bonds of DNA. This production of complementary sticky ends in DNA fragments to be cloned is important in formation of recombinant DNA. The principle is simply that if two different DNA molecules are cut with the same restriction endonuclease, both will produce fragments with the same complementary sticky ends. So they can be joined together, which make it possible for the formation of recombinant DNA. This is why restriction endonucleases are a very important tool for genetic engineering. Examples of such enzymes are ECO-R1, HIM-D3, BAGL2, SAL1, etc. However, some restriction endonucleases produce flash cuts with blunt ends, which lack sticky ends. Even flash cuts with blunt ends can be altered to make them suitable for making recombinant DNA. Examples of such enzymes are HEY3, SMA1, etc.
Using genetic engineering, scientists can create cells that produce proteins they wouldn't ordinarily produce. For example, circular pieces of DNA, called plasmids, found inside bacterial cells, can be engineered to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.